Good morning, ladies and gentlemen, and firstly and most importantly, very happy World Otter Day to you all. It is with great pleasure that I welcome you to today's webinar, and it is great to be joined by so many people from all over the world and by so many otter enthusiasts as well. So thank you for your time and thank you for joining our celebration for World Otter Day. We're very pleased to be joined by over 200 attendees that have signed up across the two webinars from around 40 countries, which is fantastic to be able to share our knowledge and share the day with all of you. So thank you for all of you for joining us. As many of you will know, IOSF launched World Otter Day to celebrate otters and most importantly, to raise awareness for their conservation around the world. Throughout the years, it's grown and gone from strength to strength, thanks to people like yourselves and people that are enthusiastic and passionate about otters. And for this, we want to thank you. This year, World Otter Day, we have events at last count in 37 different countries across the world, and it is great to see so many events happening. These range from different aspects such as education to research or fundraising for international otter conservation or for otter conservation locally in people's areas. So thank you. Just to do a little bit of housekeeping about how the webinar will work, we are delighted to be joined by 6 different panelists today, 5 different speeches, but 6 different people. And I'll introduce you to them once I have kind of gone through my little talk about how it'll work and how we will work the whole webinar today. Sorry, I'm just inviting our last panelist back in. So if you have any questions, as this is under the webinar setting and the webinar kind of Zoom webinar rather than Zoom meetings, if you have any questions, there's both the question and answer and chat box at the bottom. If you have any questions, if you put them in there, I will then ask each and every panelist that will be speaking throughout the day. During the presentation, they'll have about 50 minutes to present on their respective talks. And after that, we'll have about 5 minutes to ask any questions. Following all the presentations, depending on the time we have left, we can kind of revisit the questions, and also the panelists will have the opportunity to answer through just typing into the chat box to keep you up to date. Like I say, at the end of all the questions, we'll have an open discussion, an open chat, and thanks to all the panelists and all you guys for attending, and then we'll obviously close the session. So for today's session, we are delighted to be joined by, like I said, 6 different speakers talking on five different topics. They're all happy and here to share their knowledge, experience, and enthusiasm on a variety of different topics in relation to otters. So for this morning's session, we are delighted to be joined by Dr. Paul Yoxon of the International Otter Survival Fund, who will be talking, his presentation will be titled Otter Spraint Surveys and Spraint Analysis and Limitations. Dr. Eleanor Kean, whose presentation will be on the unexpected results from a nationwide otter survey of Wales. Mohan Shrestha, who is an otter researcher in Nepal, and his presentation will be the Eurasian Otter in Nepal, unveiling the presence after 3 decades. We have Frederick Kistner of the Karlsruhe Institute of Technology and Wild Track, whose presentation will be on what you learn by looking at otter footprints, an introduction to the footprint identification technology, or FIT, for otters. And last, and by no means least, we have Klaudja Koci and Bledi Hoxha of the Protection and Preservation of Natural Environment in Albania. And they will be talking about an overview of the European otter research and monitoring in the Prespa Lakes in Albania. So first of all, on behalf of IOSF and on behalf of everybody attending today, I would like to express my thanks and gratitude to you all for being here today. Many of you will know we have split the webinar over 2 sessions. So we have the morning session, which is obviously what we're on today. And we have an evening session, which has a focus on kind of a different part of the world. We like to do this because it helps everybody join. It has no constraints on sort of time zones or anyone that might be in the middle of the night just now. Obviously, some people may not be here for the morning session, but they can also celebrate with us later. All sessions will be recorded and put onto YouTube at a later date. So if you want to kind of re-listen or revisit any sort of topic that we're covering today, then that will be available, and all the links will be emailed out to you in time. So without further ado, we don't want to listen to myself too much longer. I'd like to hand you over to the first speaker. Like I said, if you have any questions during the presentation, if you pop them into either the chat or the questions and answers box, I will ask them to the speaker, and they can answer accordingly once they're finished. So the first speaker today is Dr. Eleanor Kean, and her title presentation is The Unexpected Results from a Nationwide Survey of Otters in Wales. Just to give you a little introduction to Eleanor. So Dr. Eleanor Kean is an independent otter specialist with a varied career history spanning wildlife conservation NGOs, policy advice, and academia. She has published otter-focused scientific papers on scent communication, diet, health, and contaminants. She coordinated the most recent otter survey in Wales with Cardiff University Otter Project and Natural Resources Wales, and she now uses her scientific training in communicating complex subjects, data analysis, and evidence review to help organizations that don't have those skills in-house. So I will pass you over to the fantastic Eleanor Kean for her presentation. Thank you.
Thank you, Ben. I'll just share my screen.
Perfect.
You can see that okay?
Yeah, we can see that fine. I'm just going to mute myself, so I'll leave it up to you. Thanks, Eleanor.
Okay, I'm just going to move you around my screen too. So, good morning, everybody. Croeso o Gymru, bore da pawb. Welcome from Wales, and good morning, everybody. So, as Ben introduced, I'm going to talk to you about our most recent national survey in Wales, and I'll explain why I describe the results as unexpected. So for those of you who aren't too familiar, Wales is part of the United Kingdom. It's one of the 4 nations in Wales, and I've put a map up there and highlighted the country in red, so you can see where we're located. We share a border with England, and famed for our dragons rather than otters. We have a dragon on our flag. And the species of otter that we have in Wales is Lutra lutra, the Eurasian otter. So that's the only species that we have here, so that's the focus of our national surveys. I'm sure many of you are familiar with the history that otters have with pollution, but just to give a little bit of background to why we have national surveys in the UK. There were big population crashes in the 50s and 60s of otters, and this was largely put down to biomagnifying and bioaccumulative chemicals, which mean otters, as the top predator in freshwater systems, had body burdens of these chemicals that were really large, and in some cases they could be directly lethal. They were at concentrations that caused death, or they were at concentrations that caused reproductive problems or immunological problems, developmental problems, all sorts of problems. They can cause cancers and so on. These persistent pollutants were then banned and removed from use, and we could track the decline of those levels in otter bodies, and this graph is from research that I was involved in doing that. And subsequently, populations across the UK improved, otter populations improved. So we had these national surveys set up. I say we, it was before I was born even, in the 1970s. And every 7 or 8 years, roughly, they're repeated to track the recovery of otter populations. So here are the maps from the 1970s through the 80s, 90s, early 2000s and 2010. And you can see as those colours get darker on the maps, more of the survey sites had signed for otters. So this was a really positive thing. And by that survey in 2010, where you've got lots of black colour on the map, 90% of survey sites had signs for otters in Wales, and at that time Rob Strachan, who coordinated that survey, he predicted that by 2020 we'd have a full recovery across the whole of Wales. And across the border in England, it was a similar picture. Otters weren't in quite the same position as in Wales. Wales had slightly better otter populations than in England, but you can see a similar pattern of the colours progressively over time getting darker as there were more signs of otters found. And so in the UK, as in a lot of Europe, otters were hailed as a conservation success story. It was really seen that the banning of those chemicals and cleaning up of waterways had seen the return of otters, and they were the sort of the poster boys of conservation, if you like. And I think, and I witnessed that this led to a certain level of complacency about the species, where people felt that, oh, you know, we've tracked this recovery and job done. We don't really need to think about otters in the UK anymore, that they're all okay. But that wasn't the case of those of us who were working in otter research or otter conservation at the time. And we knew that although those chemicals were banned, that there were other potentially damaging chemicals and other threats in the environment. And so that we didn't feel that there was space for complacency around the status of otters. And at that time I was working for Cardiff University Otter Project, which is run by Dr. Liz Chadwick. And so we continued to collect otter carcasses. So those are otter bodies from otters that are found dead, usually as roadkill, so that we could store those organs and tissues for analysis when funding became available for other chemical contaminants. And so we were asking the question, like, were otters really okay, and had that recovery continued? And at that time, we became more interested in otter diet as well. And we were doing some Spraint collections, so Spraint is otter droppings, to investigate changes in otter diet. And we noticed that there were fewer Spraint samples than there had been 5 years previously in the national survey. And so we alerted the statutory nature conservation body in Wales, which is Natural Resources Wales. And together we set about organising a repeat of the national survey across the whole of Wales. So the methods that we use is a standardised method. It's the same one that's been used since the beginning of the national surveys in the 1970s. And this means in Wales, we've got over 1,000 sites that we surveyed between 2015 and 18. So we finished this quite a few years ago now. And at each of those sites, up to 600 metres of riverbank or coast are surveyed. And we used experienced or trained volunteers for this survey. Previously, on some of the national surveys, professional surveyors have been paid, but unfortunately there wasn't funding for that this time. So we used experienced or we trained some volunteers. And it's unusual on these surveys to actually see an otter. So what we're looking for usually is their spraint, which is their droppings, or footprints. And that's what these the 1,073 sites look like spread across Wales. So the green dots are where we found signs of otters, and the black dots are where we didn't. The little red crosses are the handful of sites where we didn't manage to get to this time. So there were only a few sites that we really missed. And you can see that there's a spread, the good news is there's a really good spread of green dots. So we've got still got lots of signs in Wales. But unfortunately, when we look at it overall, we have got fewer signs than we had in the previous survey. You can see there this steady recovery from the 1970s to through the 80s, 90s, 2000s, 2010, and then we've got this drop off now in this most recent survey. And if we do that sort of averaging over the regions and look at what that looks like, in the previous survey in 2010, we had this strong dark green band from West Wales up through the middle and north. It was looking really good for otters in Wales, and we've had quite a distinct lightening of some of those colours, and really only one region managing to keep a similar colour. And so I'm going to focus in on a couple of those regions now, just to give you a little bit more detail. We've got the Severn, which borders with England, and the Tavy over in West Wales. So you can see, looking at this map of the upper seven catchment, that we've got lots of green dots, which means we had lots of otters, and that the percentage of sites which had signs for otters was very similar in 2010 as it was to the most recent survey in 15, 16 and 17. We had 94 and 93% of sites had signs for otters, so very good for otters, and it stayed very good. At the other end of the scale, the River Tavy in West Wales had very good signs for otters in the early 2000s, and in 2010, 97 and 96%, so nearly all of the sites had signs of otters. But in 2015 to 17, that dropped down to 50%, so a really big drop in the number of sites that had signs for otters there. So this is a catchment that we're particularly concerned about. And so this really begs the question, why? You know, that was at the forefront of our mind when we looked at this data, and it continues to be at the forefront of our mind, and I'm sure you're thinking it as well, looking at those now. So it's a possibility that there are chemical contaminants in rivers and that otters are exposed to, that are having an impact, and so it's important to carry on collecting otters that are found dead and analysing their tissues to check for that. Occasionally they come into conflict with humans, and this is a snare that was removed from an otter, but I don't think that's at a level that would cause this decline in signs that we saw. Ingestion of plastics globally is a concern for wildlife, and certainly that does occasionally occur in otters. There's concerns about habitat, overgrazing, grazing right up to the river bank, removal of bankside vegetation, and there's a concern perhaps around there being adequate sites for females, undisturbed sites for females to raise young, but whether that has significantly changed since the sort of early 2000s, 2010, 2020, I'm not so sure whether that's the case. And we see increasing reports of pollution incidents and pollution generally, diffuse pollution as well, in our rivers in the UK, and that's certainly an avenue that's worth investigating, and whether those pollutants are having an impact on the food chain and the prey species on which otters depend. And certainly we know in Wales, as in many other areas, salmonids in particular are really suffering, and all of the populations, all of the salmon rivers in Wales are considered in categories that are called at risk or probably at risk. And those are same for salmon and trout, and eel are similarly in not a very good position. So there's potentially a problem with food supply for otters as well. But what I'd really like you to take home from this, when you're thinking about why this might be happening in Wales, and that there's a really big amount of uncertainty, and we can't be sure at this moment. We've looked for any sort of obvious spatial patterns in the data that might give us a clue for what it might be, and there isn't anything glaringly obvious. So it really requires some in-depth investigation and not jumping to conclusions on what it might be. And it might be a combination of many factors, and it might be that different catchments have different pressures on the otter populations there. So what are we doing next? In Wales, I'm part of a steering group where we're looking at ways that these declines could be further investigated, and also planning future surveys. Perhaps we'll have the next national survey repeated in 2024, maybe around that time, and doing some interim monitoring to go back to some of those catchments to look at what's been happening, and we have completed some of that. So we went back to the River Tavy. Remember that catchment that I said we were particularly concerned about, and a professional, highly experienced surveyor went and resurveyed all of it. And again, looking at in the early 2000s and 2009-10, it was at 96%. Our volunteer surveyors found 50% of the sites had signs for otters, and when we sent the highly experienced surveyor back in 2019-20, he found 74% of sites. So quite a bit more than our volunteer surveyors found, and that's good news for the catchment, but that's still quite a big drop from what it was in 2010. So there's still a concern there and that professional surveyor said that it took him quite a long time to find signs for otters. It was much more difficult than he would have imagined for that catchment that has previously been a really great place for otters. In England, a national survey is underway this year. I think that was spurred on by our results because it had been 12 years since there had previously been one in England, and those data will add to this investigation if they find similar. And the work of Cardiff University Otter Project is really important that continues, and that contributes to investigating why there might be declines in otters. So that just leads me to say, if you want to access the full report, you can access it here on the Natural Resources Wales website. It's a bit tricky, that link, because it's in the Welsh language, so I'll pop a link to that in the chat if anyone wants to deep dive into the results in more detail. And to say thank you to my partners on this project, which was Cardiff University and Natural Resources Wales. Thank you, IOSF, for inviting me this morning, and a little thank you to Jeff Chard, who's a brilliant otter naturalist and often lets me use his photographs. Diolch yn fawr, everybody, thank you very much. And I'm happy to take any questions. If you think of any later and want to contact me, you can get me through my website as well. Thank you very much.
Thanks, Eleanor. That was a really good presentation and really emphasised the importance of not being complacent, I guess. I think when I was, from my point of view, when you're talking about why they've gone down, it does also emphasises the complexity of it, with otters being at the top of the food chain. It could be a fact if they've lost their prey. It could be a fact that any sort of pollutants that are being eaten by things further down the food chain end up in the otters, or any of the factors. I don't need to kind of repeat what you said, but it just shows the complexity of it, basically, and how fragile, especially sort of top predators, can be.
Yeah.
If anybody has any questions for Eleanor, then please feel free to just put them in the chat box or to put them in the questions and answers, and I can ask herself.
And I'm just going to put that link to the report in case anybody wants it.
Yeah, that's great. I'm just going to close this. Yeah, so I think, yeah, having another national survey will be good, and hopefully we'll be able to show a trend and maybe
to see,
more positive.
Yeah, to see if that decline, you know, either it's continued or it's leveled off.
Yeah, exactly.
Or even if it's like gone up again, that would be great if it was a little bit of a blip.
Yeah, absolutely. So there is one question for you that says, is there any research on whether reproduction numbers are declining?
So I guess what you mean by that is the number of otters reproducing are declining. I think that's the question. So Liz Chadwick at Cardiff University Otter Project, where they collect carcasses of bodies of otters found dead across England, Wales and Scotland, has had a look at the data to see if there has been a decline in reproduction. There is, I don't want to like preempt her findings because I know she's sort of in the middle of it, but there's nothing dramatically obvious. There is still signs of reproduction, so it's not as though reproduction has stopped altogether. There's still signs. So there's, you know, occasionally pregnant females or kits, or so it's not as though reproduction has stopped. And this decline that we've seen isn't a huge decline. You could see from the green dots across Wales, there's still plenty of otters, but so that's not to say we shouldn't pay attention to it, but nor is it like, you know, it's not the crashes that we saw before.
Yeah, I guess that's why the surveys are important, because you've obviously seen a decline, and then another survey, if you see another decline, then there's potentially bigger issues. I know that one of our panelists actually has a question. Fred, you can ask it yourself, or do you want me to just read what you typed? He's unmuted. Okay.
Hear me now, Ben?
Oh yeah, I can hear you now. So yeah, go ahead.
Sorry, my bad. Yeah, I had a question whether the surveys were conducted especially the surveys with the very high identification rate were conducted by professionals or by volunteers, and whether there was a chance of accidentally overestimating the number of positive sites.
So is it possible that the previous surveys were an overestimation, or ours would be an underestimation? We did consider that, and we did a bit of an analysis to try and rate the experience level of the surveyors and see if that changed the probability of the site being finding signs of otters or not, and we believe that it hasn't. That's not to say that it definitely hasn't. And I think this is what the England survey that's being conducted this year will be. The data collected from that will be really useful to assess that in more depth, because they've had funding to well, a lot of funding. We had a very teeny tiny amount of funding. Some of the work's being done by professionals and some of it's being done by volunteers simultaneously, so they'll be able to better assess that, really. So it'll be really useful to have that data coming out of England.
Yeah, because in my experience, I know there's a publication from the early 2000s on river otters in Texas from Jonah Evans, and he was looking into especially like user bias of operators and about their tracking ability to read tracking signs, and it was quite so he came up that it's quite important, and there's quite a difference even when it was trained professionals who did the survey, when they liked their special tracking signs expertise in that regard. So there was just something that came to my mind might also be an explanation.
Yeah, certainly it could be. One of the other things we did was that I only showed you one of the catchments, but we've had really experienced surveyors go and resurvey some of the catchments to see if they found different, and they found a similar
trend.
Yeah, similar trend. So it's not perfect. It's not a perfect methodology at all, and it is it's sort of a pragmatic approach, really. It is what we can do on a national scale.
Absolutely.
And I wouldn't say the percentage of positive sites is an exact amount. It's an indication of what's happening to the population rather than an exact figure.
Yeah. There's a couple of questions that are kind of linked. There's one from Elma Boyd and another one from Jane Kirkwood. So Elma Boyd has asked, will the raw sewage that is now allowed to be dumped in our water affect the otters? And then that kind of links on to Jane Kirkwood's question of our water samples being taken to identify any particular contaminants.
So sewage pollution is obviously something that's in the news a lot at the moment, and does impact freshwater ecosystems. Certainly, what level otters can tolerate, that's, I wouldn't say is not really exactly known. Something that otters, even where there are pollution incidents where lower down the food chain there are problems, or even otters are a generalist predator, so they can readily switch to other prey to sustain themselves. So it's not really clear at the moment what impact levels of sewage pollution have on otters. It's something that requires more investigation, really. And in terms of water samples being taken, it's not something that I'm directly involved in myself, but there are lots of great organisations doing work on water quality or invertebrate sampling, and it would be great if someone had the funding to put those two bodies of work together. So looking at national survey data with what's happening in the rest of the freshwater ecosystem, that would be fantastic if someone had the money to do that.
Yeah. There's a couple more questions and then we'll hand over to our next speaker. So Teresa Mason's asked if you've ever used dogs. She's a volunteer and her dog is able to find Spraint much more than she can do.
Teresa, I agree. I used I used to survey with a dog about 15 years ago, and without even intentionally training him, he just got used to me being excited when I found Otter Spraint, and then he copied that. I know there are some people who are specifically training scent detection dogs at the moment for finding Otter Spraint, and again, that would be really interesting if there was some research looking at their detectability rates against an experienced surveyor or against a volunteer, to compare those different methods. On a national scale, to do a national survey effort, I mean, even Wales, a really small nation, that's 1,000 survey sites. It would require a great effort to get enough dogs trained up to do 1,000 sites. I mean, it'd be fantastic if we could, but I think practically that might be quite difficult.
Yeah. Last question for you, Eleanor, is from Kiwi, and their question is, are there any periods which had year-on-year surveys conducted, and how much do otter populations naturally fluctuate year-on-year?
Great question, Kiwi. I would love it if there were some areas where there were surveys conducted year on year. I think that's really needed. We don't understand that well enough at all about otters, how their populations fluctuate. I wouldn't imagine that it would be quite so obvious as year on year, because think about how long lived they are and how long that the cubs stay with the mother as well. So I don't think we'd see a fluctuation from year to year, but certainly it'd be useful to monitor year to year to see how that did fluctuate. And I would love it if that existed, but trying to get statutory nature conservation bodies to even do a survey every 7 years is difficult enough. So unfortunately at the moment we haven't got something year on year, but I am in talks with some conservation organisations that hopefully we might be able to get some smaller areas surveyed more regularly.
Perfect. Well, thanks, Eleanor, and thanks for your presentation and for answering all your questions perfectly.
No problem.
Thanks. I'm now going to pass you over to our next speaker, who I will introduce. Our next speaker is Dr. Paul Yoxon of the International Otter Survival Fund. And I'm sure many of you that follow IOSF may be aware of Paul and his work, but I'll just give you a little introduction anyway. So Paul is the head of operations for IOSF, which he co-founded in 1993. In 1999, he completed his PhD on the otters on the Isle of Skye and their use of the coastal zone. He found that this was closely related to the geology of the island, as otters using coastal habitats must have fresh water in order to wash the salt out of their fur, and the availability of freshwater pools is related to the porosity of the rocks. Paul has worked on many TV programs, including Wildlife on One, The One Show on Blue Peter, and has lectured to various groups, including school and university students, natural history societies, and as part of Aberdeen University's extramural education program. He has also collaborated on several international projects, including the hairy-nosed otter in Vietnam, Eurasian otters in Belarus, and worked with the Dutch government on otter road mitigation in the Netherlands. Since 2007, he has been very active in combating the illegal wildlife trade and has organised several training workshops, and most recently in Malaysia. And I know some of our attendees today were actually either speakers or attended there, so welcome to you guys as well. And his main roles lie in research, lecturing, and fundraising for the IOSF. So without further ado, I hand over to Paul.
Good morning, everybody. I'm just going to share the screen.
Yeah, your screen share, just need to open the presentation.
Okay.
Yeah, and then just go full screen.
Okay, is that okay?
Yeah, that's perfect, thanks.
Okay, as I said, good morning, everybody. I'm here to talk on otter spraint analysis and spraint surveys, and also to look at the limitations of both. This is an otter spraint, which is composed of fish bones, mucus, small mammal remains. So what is a spraint? It's the dropping of the otter. Otters have an anal ascent gland, so they deposit a chemical code on the spraint. And a lady called Dr. Trowbridge from Aberdeen University, she did a PhD on the chemicals in the otter spraint, and she found there was 300 different chemicals. And she worked out that maybe when an otter smells a spraint, it can say if it's male or female, or maybe young or old, or even it can identify an individual. Because otters have a cribriform plate, which we don't as humans. So it's like a cavity between the nose and the brain, so they can process the chemicals in the spraint really quickly. And if you think this is maybe a bit far-fetched, we used to run wildlife trips out to Russia, and we worked with a guy who worked on brown bears, and they used KGB sniffer dogs. They could identify individual brown bears by smelling the brown bear droppings. This is an otter sprainting. This is a Eurasian otter. And this is a collection of the otter spraints. You can analyze the otter spraints and work out the diet. You can see here otters are sprainting under the bridge. This is an otter spraint near this culvert, and this is where you can collect them. Also at the junctions of a river. And this is an otter spraint. You can see the green buildup of algae. So you collect the otter spraint, and you can dissolve it in a false teeth cleaning powder, or if you haven't got that, you can use biological washing powder, and you leave it overnight, and then you sieve it, and then you let it dry out. And this is the dried otter spraint, and you can identify the vertebrae of the fish bones. We're lucky in our country, and we have guides to otter spray remains. We were out in Malaysia a couple of weeks ago on a workshop, and they don't, but you can make your own guidelines by catching the fish and identifying the vertebrae. And this is some of the fish vertebrae. And by using the guide, you can identify the fish. There's 3 ways you can sort of analyze the spraints. There's the relatively frequency of occurrence, where you number of prey items in all the spraints over the total number of prey items identified in all of the spraints, and you multiply this by 100. Or the percentage of frequency occurrence. This is the most easily applied. It's the number of spraints containing a particular prey item over the total number of spraints in the sample times 100. But when I did my research, I think I analysed about 1,400 spraints, and I also did random sampling where I basically just had an X, and the spraint near the middle I identified. That's how I did the random sampling. And when you look at the data for the Isle of Skye, you can see percentage by random sampling and frequency of occurrence were pretty much the same. I also found out that 5 key prey species, you can see the ones at the top: the blenny, the rockling butterfish, the saithe, and sea scorpion. They made up about 82% of the diet. And this was also the same in Shetland, in the Outer Hebrides, and the coasts of Norway and Sweden. So these 5 key prey species are really important for the Eurasian otter that lives on coastal areas. There's limitations. As I said, the percentage frequency of occurrence most easily applied. However, there seems to be little agreement among authors on how to interpret the results. You should also realise that in the UK, 3 times more spraints are found in the winter than in the summer. Also, you'll only get hard-bodied prey will be identified. Our otters that live in the marine environment also eat octopus, so you won't get any octopus coming out in the spraints. They also like the livers of dogfish, so you'll see a dogfish on the shore, and they've sliced down the skin and taken out the liver. Dr. David Carrs from the Institute of Terrestrial Ecology at Banchory, he did feeding trials where he fed captive otters, captive Eurasian otters, and he found that Spraint analysis overestimated the 2 proportions by 5 to 2,900%, and it could underestimate by 12.5 to 50%. Well, what Spraint analysis does do, it ranks the prey in order of importance. So that's all it can really do. So it is a useful tool. I wanted to now look at, can you estimate otter numbers using Spraint? The Environment Agency in the last survey of England, they surveyed the river Ribble, which is a river between Manchester and Liverpool, and they said the Spraint numbers have gone up by 47%, and they then argued that there was a 47% increase in otter numbers, which you can't really do. In 1986, research carried out by Hans Kruk and Jim Conroy in Shetland, it was concluded there was no correlation between sprainting and the frequency used by an area by otters. So it cast doubt on the use of Spraint surveys as a method of assessing habitat utilization by otters. So we wanted to have a look on the Isle of Skye, on the 2 coastal areas, whether there was any slight correlation between numbers of Spraints and numbers of otters. This is the home range of an otter when the habitats are at about 2.2 kilometers to coastline. This is the Ardnage Peninsula. And we did a weekly walk. We also used camera traps, and we used people who we knew who regularly monitored the coast. So we had a really quite an accurate method of assessing otter numbers. And we counted the sprainting points and the number of spraints. And you can see here, these are the sprainting numbers, and these were the otter numbers. And there was no correlation between the spraint numbers and otter numbers. And this was the spraint activity. Again, between sprainting points and otter numbers, there wasn't a correlation. And we also had a look at the Loch Nadal coastline, where we could again identify with regular walks and using camera traps, we could identify individual otters. And again, between spraint numbers and otter numbers, there wasn't any correlation. And between active sprainting points and otter numbers, there was no correlation. So you have to be very careful when using Spraint and interpreting the results in terms of diet and estimating otter populations. And I think the conventional way of doing otter surveys, where you go into a 10-kilometer grid square and you find otter evidence, all it tells you that an otter has been there. It doesn't really give you any indication of otter numbers. So thank you for listening, and if anybody has any questions on Spraint analysis or Spraint surveys, I'd be happy to answer.
Thanks, Bob. You can stop sharing your screen. I think you have actually. Has anybody got any questions to Dr. Paul on anything on the Spraint analysis side of things? If you can stop sharing your screen at the bottom, there's like a red. Okay. Yeah, that's it. There's a question from Teresa Mason here. She says, I live near a fishery and the otters don't take any carp. And they take carp, but they don't eat it all. Sorry, I misread that. Why would that be?
It's probably quite a big fish. I mean, we find that otters don't really eat the heads of fish, but maybe it's just too large a fish and it's incredibly bony.
Thanks. Is there any other questions to Paul? Jane Kirkwood asked, are there DNA studies done from Spraint samples?
There are people who are doing it, but to my knowledge, it's not been done in Scotland at all. I mean, there isn't really the funding for any of this DNA work. And since the Institute's terrestrial ecology is closed completely, they used to do most of the pioneering work. So no, there isn't anything done.
Yeah, I think Eleanor kind of covered that there needs hopefully to be more support and funding to get these kind of things going. Teresa Mason has another question, and she's asked, can you tell from Spraint how contaminated rivers are with chemicals or red signal crayfish or invasive species, I guess?
I suppose you could identify if they've been eating the crayfish, but contaminants, not unless it's the work's done in a laboratory now.
Is there any further questions before I pass over to our third speaker? No, that's great. Thanks, Paul. Okay. And we'll pass over to Fred. I'll give you a little introduction to Frederick himself, and then we'll let him take over the screen. So, Frederick Kistner of the Karlsruhe Institute of Technology, or KIT, and WildTrack. So, his presentation is going to look at kind of using otter footprints, a kind of introduction into the technology that he's working on and has done a lot of work on. So, just to give you a little introduction to Frederick before I hand over, and an introduction to the footprint identification technology, or FIT, as I'm sure he will probably refer to it as. So FIT is a non-invasive monitoring method that combines traditional tracking with the state of the art data analytics, and enables to differentiate species, sex, and individuals by only using images of footprints as a data resource. Frederick Kistner holds a master's in environmental science from the University of Landau and sees himself as a hybrid of a tracker and a data scientist. He is a founding member of the WildTrack Specialist Group and has been tracking coastal otters in Portugal for more than a decade. He is a full believer in non-invasive animal monitoring and thinks that recent developments in machine learning and artificial intelligence can further boost this approach. Furthermore, he is based in Karlsruhe, Germany, where he is currently doing a PhD at the Institute of Photogrammetry and Remote Sensing at the Karlsruhe Institute of Technology in Germany. His studies are currently funded by a scholarship of the Deutsche Bundesstiftung Umwelt. Apologies for the pronunciation, Fred, but I'll pass over to yourself. Thanks.
It's all good. All right, just let me quickly share my screen. Can you see the screen in presentation mode?
Yeah, we can see that fine, thanks.
Okay, perfect. All right. First of all, thanks, for inviting me and giving me the chance to talk a little bit about my work. As I said, I'm a PhD student at the KIT in Karlsruhe, and I'm a member of WildTrack, which I'm going to talk about a little bit more in a minute. The main topic of my talk today is about what you can learn by looking at otter footprints, and then I'll give a briefly introduction without going too much into stats about the footprint identification technology for otters. So the main part of my talk will focus on tracking otters and give I'll run through a quick overview of some things what you have to look out for when you want to track them, and then in the second part I'll go a little bit more into the computer science part, which I'm sure will be too boring, so I'll try to keep that as short as possible. As mentioned before, I'm a member of the WildTrack specialist group, and we at WildTrack, we're a loose cooperation of conservation biologists and also technologists, and we have projects all over the world. So what I'm gonna talk today about otters can similarly be applied for rhinos in Africa or tigers somewhere in Southeast Asia, for instance. And yeah, so our method, the footprint identification technique, that combines two approaches of actually looking at animals. It all started in the 1990s with Sky Alapai and Zoe Jewell, who were working with rhinos back then, and what they did was radio color studies with rhinos, I think in Namibia, if I recall it correctly. And two observations that they made when they were working in Namibia was, first of all, that radio coloring rhinos had actually a negative rate of the negative impact of the fertility rate of the species they would like to protect, and secondly, that the local guides they were accompanied in Namibia, they were sometimes laughing at them about the heavy equipment that they were using and why they made things so complicated, because for them it was all obvious. They just looked at the footprints, and sometimes they were just able to tell them which rhino to expect at the waterhole, what's the individual, what's the sex, and actually what happened. So just looking at the tracks could tell those experienced trackers a whole story of what happened maybe the night before. And so they were really impressed by the skill of their trackers, and they thought, well, if trackers can identify individuals and can identify sex, they thought, well, whether there's a method to sort of a scientifically replicable, robust method that can extract this information. And in fact, they came up with a solution, and now they published on a wide range of species, and my research mainly focuses on otters adapting the footprint identification technique. But yeah, so just a brief overview, and I go into stats a little bit further down the line. But since footprints or images of footprints are our main data resource, I thought I'll talk a little bit about tracking otters in general. And I want to start with some anatomical details. Like, otters are part of the weasel family, and if you want to look at, or if you look at footprints of weasel family and you find perfect footprints, you expect to see 5 toes. On front feet, you often see like 3 different parts of a footprint. So first you have the 5 toes, then you have the metacarpal pad, which is sort of, in tracking books, it's called croissant shaped or triangle shaped. I'll just see if I can get the laser pointer out. So, and this triangle is quite characteristic for otters. And then you have the carpal pad over here on hind feet. It's called a metatarsal pad, which is, in my experience, a bit kite shaped. And so depending on which species you're working on, like my main experience are Eurasian otters, but I've just recently submitted a paper where we looked into footprints of also smooth-coated and short-clawed otters. And you have the with similar characteristics in the front feet, which is sort of like three parts of a foot, and the hind feet, which are more sort of like can be divided in 2 parts of foot. But every one of you who has ever looked into tracks in real life might have realized that they often don't look as much as perfectly as they're all illustrated in the tracking books. So in my experience for otters, the first toe is often missing. And when you read in tracking books that you see the skin in between the webbings in between the toes, this is, in my experience, only barely visible, or as long as you're only tracking in very good substrate or deep substrate. And this leads me to the next slide. If you think of a track, there's various aspects that sort of have an influence on the shape of a track in general. And on the top left, I included this image of the recent otter workshop in Malaysia that my colleague Larissa Slaney kindly provided me. And as you can see, Ben and Larissa are kind of stuck in the mud when they were looking for footprints. And depending on what kind of substrate an animal or a human is walking on, there's this difference in their gait, but also in the shape and the texture of the footprint in general. So, for instance, in mud, my experience is that footprints tend to appear a little bit smaller than there are, because when the animal is sort of like lifting their feet, the mud is sort of condensing again. While the opposite extreme is when animals walk over dry sand, that edges and shapes tend to widen because the substrate can't really hold the footprint. So sometimes they appear a lot bigger than they are in reality. Also, the weather can have an effect. Weather like wind and rain can sort of change the appearance of a footprint, and then sometimes it can be quite challenging to identify, and only at the species level, not talking about further. And last but not least, snow, which is, in my opinion, among the most complicated substrates to work with. Even though footprints can appear very perfectly, there's so many variations, and with the melting process, it's not necessarily making it easier. In my experience, the best substrate to work with is sort of moist sand. That gives the most detail out of a footprint, while it's still fairly replicable in my opinion. Yeah, so that leads to the next question of where can you find footprints? Obviously, this really depends in the region you are working in. In some regions, tracking might not be the most appropriate method, and in other regions, it's really great to track otters. So, for instance, I did most of my work in southwest Portugal, where it's all sandy substrate and like coastal dune habitats. So you'll find a lot of good footprints, for instance, at riverbanks when they're sandy, or at dry ponds, or in the intertidal regions. I also have done some work with the German Otter Foundation in Hankensbüttel in Lower Saxony, where we did some classical bridge sampling. And if there's certain bridges that you end up and have an interest in, then it might be worth spending some extra time and just preparing some certain areas where this is a rock that frequently gets visited by otters, and I want to learn a little bit more about their footprints. It might be worth to do some laboring and leave out some sand. And yeah, if you're lucky, you'll end up with otter footprints. But if you do that quite regularly, you end up with a lot more footprints, and then there's others. So, and sometimes it's not so easy, especially when you're starting, to distinguish between otters and all the other footprints. So just as a quick exercise, if you have a big screen, can you see the otter footprint in this picture here? I'll solve it in order to not waste too much time. It's on the right hand side, and everybody who has thought, well, it's obvious, most clearly the one in the middle. The one in the middle is actually the one of a stone marten. It's very similar shape, just a little bit smaller, and if you have perfect substrate, you see more heavy feet and no webbings. But there's also badgers that, depending if you see the claw marks or not, that can be confused. Raccoons in Lower Saxony, if you have raccoons ready, is definitely a species that tricked me more than once. And down in Portugal, so we have the same exercise again. You can have another look at and think for yourself whether you spot the otter footprints. It's those over here, again, far from perfect shape, far from what you see in the tracking books, but still characteristics of an otter in my point of view. Up here you have mongoose tracks. Cats can be confusing if you have a double imprint of a cat track. It happened to me more than once that I thought, oh, cute, tiny otter, maybe juvenile, and then on the second look, I realized that there's two loops in the cat footprint, so this can definitely be confusing. You have a fox on the top right, and what, especially when I started, was maybe the most confusing, or could have been, was dog prints, because especially hind feet of otter, if you don't see the thumb, they share a lot of characteristics to a dog of a medium size in my experience. So it takes a while to sort of differentiate of what kind of animals you have there. There's more images I could put up here. Nutrias, if you have nutrias present, can be a bit confusing, though their fingers are longer, and almost any species of the weasel family. All right, so once you're able to identify tracks, it's kind of worth to lift up your head, and then not only to focus on single tracks, but if you have good substrate, you might be able to follow the tracks. And again, if you think of an animal left the track behind, and if you follow the tracks just long enough, it will most certainly, if you're able to do that, lead you to an animal, which then can be quite intimidating, because it will tell you a story of what the animal has been doing over the last hours or days of his life. And trailing is technically the science or the art of being able to follow an animal. And if you follow an animal, you might be able to find their holdouts or thick vegetations where they might spend the night. And you also might find spraints, because, for instance, if you look at the image over here where you have a pristine beach down in southwest Portugal with huge rocks and coastlines, it might be quite challenging to actually find a little rock where the spraint is on. So if you're able to follow the tracks, it might lead you to spraints, but also if you're able to identify a sprainting site, it might lead you to tracks, and it's a in between of both that can give you more insight about the ecology of the species that you're interested in. Just two remarks in regards of trailing. You need to be aware that you don't want to disturb the animals, so I always recommend to trail backwards. So this gives me the least chance of accidentally stumbling into the animal. And you also need to be aware of whether you're allowed to, whether you have the allowance to go through off road when you do the trailing, because again, you don't want to disturb any animals in any nature protected areas or national parks. So you should always consider whether it's a good idea of what you're doing right now. But if that's the case, trailing might also lead you to finding better quality footprints that you can then use for analysis, which I'll come through in a second. And one thing that I experienced can be super challenging, especially when you track otters at beaches, is to distinguish otters when you have a lot of noise, when there's several footprints of other animals, when there's been dogs running around either the riverbanks or the beaches, when there's foxes and humans. It's just a lot of distortion and a lot of noise, and something that made it easier for me to be able to follow otters is sort of to look at the characteristic gait of otters, which is called a 3x2 loop. So it's usually you have a front foot, then you have a front left, then you have a front right, then you have a front left and the right hind foot on top of the front left, and then you have the left hind foot behind. And that characteristic pattern is sort of multiplied over several regions. And even if you don't have the perfect footprints like you have in the image over here, even if you just have some dots in the sand that have been eroded by the wind or anything, that pattern, once your eye adjusted to that, you can actually follow otters for a long time, given that you have a suitable substrate. And I think my personal record is more than 6 kilometers down in a coastal dune area in Portugal, where I'm very, very fortunate to be able to track and trail otters for long distance. And I use tracks to collect data, and then with this data, with this footprints, I can extract a lot of information, and that's the footprint identification technology. Like I mentioned before, WildTrack has developed this for a wide range of species, and in my PhD, I'm trying to develop this for Eurasian otters. And the goal is to use footprints to classify species, to predict the sex, and ideally even to identify and re-identify individuals. And if you're able to do that, then to use that to do population estimates of that. So, and how is it done? Well, usually we, like I mentioned before, we use machine learning, or technically supervised machine learning. And in supervised machine learning, you teach a machine to identify patterns or to identify patterns within your data. And in order to do so, you need to tell the machine labels. And in order to do that, we cooperate a lot with zoos or other holding institutions like the Otter Center Hankensbüttel, for instance, and we collect footprints of known individuals. And we do that for many individuals to generate an image database. And with that image database, there's two routes that you can follow. If you want to have a high classification accuracy, if you want to have a better output, then the route is usually setting geometric profiles, so setting up landmark points at defined areas. And with these landmark points, you can extract hundreds of measurements consisting of distances, angles, areas. And these measurements can then be used for machine learning models that can classify species. So we just submitted a paper where we looked into differentiating between 3 different Southeast Asian otter species. You can predict sex, which I'm just currently working on a paper, but it looks kind of like it's working quite okay for Eurasian otters, or at least for Eurasian otters where I have experience in. And then identifying individuals, which is also possible, though for otters is a bit more challenging than for bigger animals like tigers or rhinos. Yeah. And there's a second route you could go, and this is also part of active research. You could use artificial intelligence, like deep learning methods for computer vision that aims to get the human out of the loop, because setting those landmarks requires quite a bit of expertise and can be quite a bit of a boring task as well, because you sit in front of a computer, you try to figure out what's part of the footprint and what's part of the background, and then you set your landmarks. You try to be as precise as possible, but also it can be challenging. It's not something you learn within 5 minutes. And so ideally we train, the ones of us who have the most expertise, we try to train artificial intelligence to get that boring part out of the loop so that we can focus more on tracking and trailing, and then get this data. All right, so my last slide, just some general overview of FIT. FIT is a non-invasive from the ground up tool for monitoring endangered species. It combines traditional tracking with state-of-the-art computer science. Big benefits are it's non-invasive, it's cost-effective, and it can be integrated in wildlife monitoring studies. For a wide range of species, we published papers showing that it's able to predict species, it's able to predict sex, it's able to predict individuals in age class, and it has very high cross-validated accuracy rates. And it provides knowledge on population numbers and the distribution of endangered animals, and this can hopefully help to be used for develop effective conservation strategies. And a big part of it, it's sustainable. So local people can be trained in the footprint collection, and therefore there's no need for constant external presence, which is often needed for more high-tech computer monitoring techniques. And this is really important to our group, that we can technically provide something that can be done continuously by people who are there. So in order to have the limited resources that are available in conservation, like ideally pinpointed them effectively. And yeah, that's pretty much it from our side. You can find out a lot more at 2 websites. So WildTrack ML, this is the computer vision and artificial intelligence project that we have. This is also a website where you can upload images in case you have already a data set, or you're interested in uploading some of them. We will soon publish an app which will make image uploading a lot easier. We're currently at beta testing phase. And also wildtrack.org, which is more a general background about WildTrack. All right, that's it. Any questions?
Brilliant, Fred. Thank you so much for your presentation. I think your presentation definitely showed the differences in finding footprints. You get to walk along the nice beaches of Portugal, whereas myself and Larissa had to waist high in mud in Malaysia. So
I have the muddy places in Portugal as well, but the beaches are definitely the joy of my work, I would say.
If you could just stop sharing your screen whenever you're ready, and I will ask a few questions that have come on the chat box. So Ben, not myself, a different Ben, has mentioned, not a question as such, but he said, really nice to have an expert confirm that dog tracks can be confusing on poor substrate. I thought it was just me.
Oh no. I
Um,
just went through some data of the. All right, one thing at a time. Here you go.
Perfect, thank you.
Yeah. So I don't know. I just went through some old data, which is about 10 years old, and I realized that I at one time processed badger footprints and mongoose and thought they were otters. So that's also something that made me realize tracking, even though it's a very old skill in mankind and especially in the hunting community, it's not what's often taught at universities, and it can be very confusing because when you look at tracking books, you see the perfect footprints.
Yeah.
But when you look in reality, they just look fairly different than that. So I could just encourage you to go out, make observations, and in my learning, something like nature awareness schools or wilderness schools, they usually have quite good tracking experience or can teach that.
Yeah, definitely. And I think about halfway through your presentation, you mentioned that when you're in Portugal, it's difficult to find Spraint, but finding footprints is easy. Whereas, for example, us in Skye, the substrate is almost non-existent. They don't often use the few beaches we have. So finding Spraint is an easier option than finding footprints. So it shows how having different techniques is really important.
Oh, it really depends on where you are. There's certain areas in Portugal as well where it's just rocky, where I look for spraints and then I hardly find footprints. Or if I'm interested in their footprints, that means a lot of laboring, me carrying tons of sand from the beach to a certain area where no otters are active. But so you can do that, but then again, it really depends on your motivation of what you want to find out and how much time and money you're able to invest. And
Yeah,
for some areas, FIT is a great method. For other areas, I'd highly recommend using DNA methods if you have the funding for that, because it's just easier. It really depends on where you are and what your research question is.
Yeah, exactly. Teresa Mason said, you mentioned background. Do you use that to identify otters too? Is tail drag useful in giving you info?
Oh, sorry. I mentioned background. Background. I mean, tail drag. Oh, that's something you see with otters, and that's something that definitely leads to disturbance in the footprint. So it really depends on your question. If you want to identify species tail drag, I've seen it in otters, but I've also seen it in beavers. So, but yeah, beavers and otters, again, can be confusing, though I don't think it's too hard, really depending on the quality of footprint.
Yeah. There's another question which I kind of know the answer to, that firstly says, thank you for this interesting talk. And then goes on to say, I would like to know if this app will cater to data pertaining to all the otter species or just Eurasian otters.
Oh, well, it really depends on my funding. I got a scholarship for another year where I really need to finish my PhD. And then afterwards, I'd be very interested in expanding this to African species and to South America. It's just about a matter of time and funding, but the interest is there. And I think the possibility is also, it should be possible to do so. And I'm hoping,
At the moment, you and Larissa are currently developing the Asian side as well. So the 4 species in Asia, the hairy nose, the smooth coat, and Asian shortclad, to compare with the Eurasian.
Yeah. So it really depends on the resolution. So for instance, to give you an insight, ideally you want to compare footprints in similar substrate, especially when you want to do the artificial intelligence classification, because if you have it in a background, if you want to do it in an optimized way, you have to be careful that the computer doesn't learn the substrate other than the geometric of the footprints. If you do it by a human operator, you can definitely avoid that, because you set the landmarks differently or similar, that you don't get disturbed that much by background substrate. And if you want to do species classification, it's roughly a good idea to have at least 100 footprints per species, ideally from a wide range of individuals. So again, you need to be careful that you don't have too few individuals, because otherwise the computer might learn to differentiate between individuals and you think that it's differentiating between species. And if you want to do something like sex classification, also I recommend to have 20, so 10 per sex, footprints of 10 individuals per sex at least, ideally. And then if you want to do individual classification, that's the most challenging part. And like species classification and sex can be done by single footprints, while individuals, you need trails of footprints. So you need 5 to 7 footprints of the same foot with good quality, and that requires more training and more effort to put into. But if the data is there and if the time is there, in my opinion, it can be done. And the better the images of the footprints, the better the quality of the footprints, the more I trust the models. Some of my research is also looking into what to do with imperfect footprints. So I'm working with statisticians of sort of like looking into imputation techniques that you can only use fragments of a footprint and then sort of model the rest of it. But it's still ongoing research. So.
Brilliant. Kiwi's asked, you mentioned stone marten. Does the AI program account for mink tracks too? And are their gaits different to otters?
I don't have too much experience with minks, to be honest, but I think if I recall it correctly, minks are in the weasel family, and within the weasel family, those kind of loops are quite common to cover some distance. So also a different gait is really depends of what an animal is doing. So if an otter is just going out of the riverbank, it probably won't travel in the 3x2 loop, 3x4 loop. It will more go in a small walk, sniffing around, maybe sprinting, then going back into the water. But if it wants to cover some distance, then those 3x4 loops are among the gaits that I've experienced the most. And Eleanor just asked a question regard of sex classification, which is a very good question. And can it determine between young male and adult female? I think this would be so it definitely related in between with the size, and I think this would be a potential source of error. But we don't only classify on simple measurements like length and width of the footprint. Like from a single footprint, I extract more than 200 measurements. So I would assume that for that even younger male might have different characteristics and this is just hypothetically, I don't know, I haven't looked in that data specifically, but something like more muscular toes, for instance. So, but again, it depends on the demographics of your data set, and this can be a challenge. So ideally, I think it will work better with adult animals, and then juvenile animals can definitely be a source of error.
Yeah. There's one question, you may have seen it, about can you show the website again? Would you be happy just to put
Sure,
I
just put the links in the chat, and then
Fred will put the links in the chat soon, so anyone that's more interested and wants to look a little bit more can have a wee look and kind of show their work in more detail. Before I pass over to our next speaker, does anyone have any final questions for Fred? Give you a few seconds to type. Perfect. Any final questions that you might have, you can put it into the chat box and we can either ask at the end or we can answer kind of during the other presentations. Thanks, Fred. And
thank you for inviting me.
Thanks for the interesting presentation.
Thanks for setting this up. Yeah.
No problem, my pleasure. So I'm going to pass over to our next presentation and our next presenter, who is Mr. Mohan Shrestha, who is an otter researcher in Nepal. Mohan's going to present on his work on the Eurasian otter in Nepal and unveiling the presence of the species after 3 decades. Just to give you a quick introduction to Mohan himself, Mohan is an environment and wildlife conservation professional and educator. His current involvement is in wildlife research, primarily on otters in Nepal, and he advocates on environment and wildlife conservation, education, awareness, and conservation policy implementation in Nepal. So without further ado, I would like to hand you over to Mohan for his presentation on Eurasian otter in Nepal.
Yeah, thanks, Ben.
Hi.
So I'm sharing my slide.
Perfect. Yeah, once you go full screen, we'll be able to see that perfectly.
Okay, is that okay now?
Yeah, that's perfect. Thanks, Mohan.
Yeah, thank you, Ben, for this opportunity. Like, we are just in the beginning of the otter research in Nepal. So I hereby trying to speak on a few evidences that I have collected during my researches in past 5 years. So it's like I would like to begin with the otter species in Nepal. Like in Nepal, we have 3 species of otter. One is Eurasian otter, and other is smooth-coated otter, and third one is Asian small-clawed otter. These are 3 species that is present in Nepal as per the records. So regarding the distribution of the Eurasian smooth-coated otter, like we need to go through the geography of the Nepal. So Nepal is basically in 3 geographic regions: the southern flatlands and the mid hills at the middle part, and north in the Himalayas. So usually, like smooth-coated otter is present at the flatlands, and you see all these red dots or bars where it is reported as present in Nepal. So this is about the smooth-coated otter. And regarding the Asian small-clawed otter, so after 1839, the distribution and the records of otter in Nepal is yet not confirmed. So it is still data deficient in Nepal. We need to work a lot to collect the evidences to show the presence of it in Nepal. So here is my presentations about the Eurasian otter. So this is the one otter, like globally status and near national status, it is regarded as a near threatened in Nepal as well. So it is said, it is one of the widely distributed otter species in Nepal, from the lowlands to the hilly areas and to the mountains. So you see all these red bars showing the distributions of otter in Nepal. But say, unfortunately, the presence of otter, though it is said widely distributed in Nepal, since 1991, there was no records of presence of the Eurasian otter in Nepal, maybe unfortunately due to the lack of the studies. So after 1991, there are no records. But after my studies in recent study in 2020, we've been able to record Eurasian otter presence from the 3 different districts of Nepal. So one you can see with camera trap images, and other is direct sighting, and other is from the carcass or the skull presence in the other part of Nepal. So these are 3 different reasons where I've been able to gather the evidences of its presence in the Nepal. So besides this, so another recent research in the Pelma River in Western Nepal, I've been able to gather the evidences like otter pelts that was killed unknowingly during due to the entangle in a fishing loop. So you see in the picture, toe with the otter is entangled in the fishing loop. So it was unfortunately killed. So this is also the presence evidences from the Western Nepal. And the last, the most recent is the findings from the West Seti River. Again, it is from the Western Nepal, where we've been able to capture a camera trap image of the Eurasian otter from that area, showing the presence evidences in the Nepal. So altogether, in these 2 years of study in the otters, in Eurasian otters in Nepal, I've been, and my team, been able to capture, gather the evidences of Eurasian otter presence from 5 different districts of the Nepal, from the middle to the western part of Nepal. Still, we've been working to gather the evidences in the eastern part of the Nepal. So we have still more to go, because it is said that we have the 6,000 rivers in the Nepal, and the records that I have shown is from only the 5 districts and 5 rivers. So still more to go. And other is, as I said, we are just in the beginning of gathering the distributions and presence evidences. We still have more to study on the impact studies, because all the infrastructure, especially the road networks, are constructed alongside the river banks, which are the habitats that otters share with the infrastructure. And another one is like we've been developing lots of hydropower. So we dam a river where the otters live. So that could be the more other impacts study that we need to go for the detailed study. And another is like we've been gathering the illegal otter pelters. That's in some from 1989 to 2017. I've been through 24 incidents. I gathered the 755 otter pels trade seized in Nepal. So basically it is said Nepal itself is not the importer or exporter, rather than, it is like a transition point where the otter pels trades from the India and then to Nepal and goes to the northern border like China. So still we have a lot more to do with the population studies as well. So I thank to all these my team members. Without them, I can't get all these evidences. And thanks to my mentors. And finally, the supporters like IOSN SSC and IOSF. So these are some of the papers that I have recently published. And thank you for your attention. May there be questions. I'll be happy to answer. Thank you, Ben.
Thank you, Mohan, and it's really interesting to hear about the work, especially your work finding Eurasian otters after 30 years, because there was obvious concerns whether they were still there, and obviously your work has found that. I have a question in regards to the Asian short-clawed otter. Obviously, you mentioned that they haven't been found for a very, very long time in Nepal. Is it likely that they're still there, or is it hard to say that?
So you mean like Eurasian otter presence in Nepal?
No, the Asian small-clawed otter?
No, yeah. I think there could be presence because we still have more to do. We do not have this study as it has been in other part of the world. So I think there must be present, but we need to go for this study.
Yeah, absolutely. Yeah, it's very, very important, especially after they haven't been found in many years, hundreds of years. So it'd be good to try and find them. But finding the Eurasian otter and doing that sort of information is gaining that information is great to see as well. Does anybody have any questions for Mohan on his work, or any sort of questions about otters in Nepal? He has a lot of knowledge about all species in the area as well. You got a lot of comments saying good presentation, congratulations on your findings and all that. So I'll give them a few more seconds to see if anybody has any questions about your work on the areas. And is it something you're going to continue researching, the Eurasian otter in Nepal?
Yes, Ben, I've been these days, my study in the Western Nepal, West City is under the progress. I have visited once, so I'm thinking for next visit, but I'm still waiting for some funds to be gathered,
Yeah. So there's a question which says, is there historically much interspecies competition between the three otter species? So is there any conflict or competition between the Eurasian otter, the small-clawed otter, and the smooth-coated otter?
Most probably there could be, but still I already mentioned we need to go for some detailed study. We right now we've been able to gather just the presence and its distributions only. So I think there could be some interspecies competitions between the smooth-coated otter and the Eurasian otter, because the habitat I showed in the distribution, I think it overlaps with each other. So there could be the possible, but we need to go for this study.
Yes, absolutely. And Teresa Mason's asking, do people support your work, or are there conflicts with, for example, fishermen or fishing?
Usually, actually, there are not more evidences of conflict between fishermen and the otters. But Nepal is highly geographically challenged terrain, and people go for fishing for their subsistence living. So it exactly people do not exactly go for kill the otters, but instead unknowingly when they set the fishing loops, it is unknowingly trapped in that fishing loop, and they are unfortunately killed. So as per my knowledge, I think there are very low conflicts between otters and the fishers.
Oh, that's great. So any sort of deaths caused by fishermen is generally an accident? It's not?
Yes,
Yeah.
yes, I think it's generally for accident.
Oh, okay. Yeah, that's good, I guess. It's good. So that if you're engaging with fishing communities, do they like the otters or?
Yeah, of course, like they are the one who provide me and who helped me a lot during my study. Without them, I could not gather all those informations. And they have a pretty, a soft corner for that the species survival as well. They used to say that the otters were found or they see quite often during the last decades and like past 20 years. But nowadays, I don't know, like they say the number of otter sightings in their locality are decreasing. And that could be, there are different reasons that could link with, could be the climate change, habitat parameters, and some other issues as well. So we need to find all those solutions as well.
Yes, absolutely. I'm just going to give people a few more seconds, see if they have any further questions. And if anybody has any questions throughout our last presentation, then we can ask Mohan or any of the other presenters at the end if you have, or they can also answer themselves within the chat or questions and answers box. Just another great talk. Thank you and good luck for the future, Mohan. That's from Kiwi. Well, Mohan, thank you very much for your presentation. I will pass. Thanks, Ben. My pleasure. I will pass over to our final presenters, who are Klaudja Koci and Bledi Hoxha. They are based in Albania, and they just gonna get my little sheet of paper to make sure I read that out properly. So they're going to present today on an overview of the European otter research and monitoring in the Prespa lakes of Albania. So I'm just going to give a little introduction to both of them, and then I'll pass over to Bledi. So Bledi Hoxha was born and grew up in southeast Albania. From 2003 to 2008, he studied biology in the Faculty of Natural Sciences at Tirana University. He joined the PPNEA team in 2009 as a volunteer, and since 2013, he manages the Balkan Lynx Recovery Program and has 10 years of experience in the research and monitoring of lynx, bear, wolf, and golden jackal in different studies in the Albanian regions. He is a co-author in different articles on large carnivore ecology and conservation issues, and recently he has had experience as a mentor on wildlife research and monitoring methods to the local NGOs and GOs in Albania, as well as the foreign interns involved in the BLRP project. Klaudja Koci is from Albania, and she is an outdoor educator by degree with a master's specialization about 3 European models of education, culture, and societies awarded by the European universities of England, Norway, and Germany, a joint degree MA Transcultural European Outdoor Studies. She is working with the Protection and Preservation of Natural Environment in Albania, or PPNEA, the oldest nature conservation in the country. Since 2014, Klaudja got involved as PPNEA's officer for the transboundary environmental network called Prespa Net, established together with 2 other environmental organizations based in Greece and North Macedonia, and is currently managing the Prespa project Biodiversity Conservation in Transboundary Basin. She also coordinates the education programs among children and young people in and around some of the most important protected areas in Albania, and hopes to instill passion for wildlife species and habitats. So thank you again for agreeing to present, Klaudja and Bledi, and I will hand over to yourselves. And we can also see your screen. So thank you.
Thank you, Ben, for the introduction, and thank you for inviting us. I am just going to share with you a little introduction on our work and our presence in Albania throughout the years. And my colleague Bledi will be sharing with you more results, knowledge and updates on the otter research and monitoring in Albania. So as you mentioned, Ben, we are a national organization focused with work in the entire country of Albania. We just provided here a map so you can actually see some of the areas where we are located, which is pretty much from the north to the south and then west east. The work that we're doing is focused in protected areas. It is about declaring protected areas as well as working towards effectiveness of those that are already declared. Also, we're focused on working on Natura 2000 habitat mapping and working in important key biodiversity areas or important bird areas and Ramsar sites, wetlands. The NGO was funded in 1991, so it's 31-year-old, and we're celebrating that over the next month. Our work is focused for the upcoming 4 years with a new strategy in 4 programmatic areas. So it's threatened species, conservation, protected areas, promoting sustainable development, environmental education, engaging, strengthening environmental organizations from the country, and of course, always advocacy and lobbying. More specifically, Prespa is an area that is of primary interest to us. We have a local office that was opened 2 years ago through the great support of Prespa Nature Trust Fund. And the Prespa Lakes area is an area of international interest. It has just been declared as a Prespa transboundary park, is bordering 3 countries, Greece, Albania, and North Macedonia. And it is composed of 5 protected areas, 4 of them national parks with the highest status in our countries, and a nature park. Some of the other governance levels that the area is exercising or like is part of, or is the fact that both Prespa and Ohrid that you can see here in the map, they also are part of a transboundary biosphere that was declared back in 2015, are important Ramsar sites and key biodiversity areas. For our work here in Prespa, which has been going on for many years, for almost 25 years, we have created a network of 3 environmental NGOs for Prespa. Bledi, if you can help me with the next slide, please. And we have created this network of PPNIA from Albania, MES from North Macedonia, and Society for Protection of Greece. We call it PrespaNet, and we are undertaking joint actions in the area, joint environmental actions, since 9 years now. This is an overview of the Prespa Lakes, where we have the big lake, the Great Lake, and the small lake, and 2 important islands. One of them is also very well known as the Snakes Islands, and is a place of great interest for research, for researchers and other institutions. Yes, please. And then with PrespaNet, Bledi, please. Yeah, maybe it's going a bit slow on your side. Then with
I'm trying to change the slide, but it's not working. Fortunately.
Okay, sorry. Okay,
Bledi,
I can share it.
Yeah, that's great, thanks.
And then another one here, please. So to sum it up on our side, this is from my side, sorry, this is a photo of our local office. It is a field station with a capacity of 4 to 5 people staying over. We have envisioned it as a place for students, interns who could come and spend some time to research the area, like weeks or months. So we're having usually Erasmus students staying over, and we're using it for ourselves. And then specifically with the work for species, the otter research and monitoring work is part of the species program that we have as part of our Prespa project. Is one of the 4 or 5 other species such as bats, chamois, a brown bear, and the work that is going on for birds as well. I will just hand over to Bledi for his part. Thank you.
Thank you, Klaudja. So this is it's just a slide that just present or give information about the main partners and donors that support PPNA about the mammals research and conservation in Albania. So yeah, I will start with with the case that we done a survey about the otter in Prespa National Park in Albanian site. And before to present what we have done and the results and conclusion, so I would like just to give an overview about the otters status in Albania. So the first data on the otter in Albania were provided by Pirgon in 1986, and in their paper the authors concluded that the otters were widespread in the most country and has healthy populations were localized in the north west of Albania mostly, and the south part of Albania. And this is the map that show all the rivers and lakes in Albania, and other research has been done in some part of Albania, for example, in 2008 and 2010. So research was being done about the distribution and status on and diet of the otter along the Drina Valley, which is localized in the south part of Albania. And later other survey has been done in other rivers like in Alsumi, Semani and Devoli, which is localized more in the central part of Albania. And in 2012, in the study that has done by Bego et al., it was focused more to estimate the density of otter population in both lakes of Prespa, in Macro Prespa and Micro Prespa. And based on his study, it concluded that the density of otter in Macro Prespa was optimal compared with the result that he got it from the Micro Prespa lake. And later in 2017, 2018, PPNEA did a survey in the main three lakes of Albania, Shkodra, Ohri, and Prespa. And the aim of this survey was to estimate the presence of otter in three lakes. And based on this study, the results show that the presence of otter was higher in Prespa Lake compared with other two lakes, Shkodra in the north and Ohri, which is nearby to Prespa lakes. And in last year, we did another survey on otter presence in Albania, and especially in Narta Lagoon, with the aim to identify the potentials as a potential site of Natura 2000, and when the country will be in of the EU. The site has been listed already, and it's a candidate one to be site of Natura 2000 network. And yeah, the presence of otter was quite good in all lagoon, Narta Lagoon, and again we did a survey on otter in Prespa, in both Prespa lakes, with them to estimate the parameters of otter population in Prespa lakes, and to increase the capacity of Koci Rapa staff, because this protected area has been is managing by the Rapa Agency of Protected Areas, Regional Agency of Protected Areas, because the protected areas in Albania are managed by the National Agency of Protected Areas, and there are 12 regional or protected Agency of Protected Areas in different regions in Albania. So we conduct this study with the Rapa staff in Koci region. So first we train them how to collect the data, and after they collaborate with them to collect data in the field. And the most important part of this survey is that this data will help the administration to prepare a good plan how to protect otter, and to do a good management inside of the protected areas related to the mammals, and especially for the otter. So both lakes in Prespa Basin are very important for otter, because contain important habitats for otter that can live and reproduce. And in the picture A, so there is a view that shows the macro Prespa, a part of macro Prespa lake, and you see the habitat is completely different with the micro Prespa lake that show in the picture B. So there is a rocky vegetation cave habitats along the macro Prespa lake shoreline, and in the micro Prespa we have red beet habitat, and it was covered with vegetation, and in somehow the habitat is good for otter, but still otter is limited to move or to catch a prey because it's quite dense. And we think that the best habitat for otter are localized in macro Prespa. And we use the two methods to estimate the parameters of otter tent population. So the first method, and the main one, is the Spraint survey that we did in along shoreline in both Prespas. And as alternative method, we use a camera trapping survey. So based on this method, we collected data during September and October last year. And the otter field work was conducted by PPNEA team, Klaudja Koci, and interns. So we had 2 interns at this time. And we defined in total 25 stations, 28 stations, 25 in macro Prespa and 3 in micro Prespa. And the short distance between each station was 1 kilometer to 3.5 kilometers. And the minimum of transect was from 200 meters to 600 meters. So this is the map that shows where the station was defined and observed by us. And so you can see there are, the stations in the macro Prespa lake and in the micro Prespa lake, and the transect as well that we did within the one station. And the camera trapping survey method, we set 3 cameras, but we investigate 6 caves in macro Prespa. And based what we estimate that 3 of them were very important to set cameras, because the caves was used as a shelter by otter. We found spraints and very fresh spraints. So we decided to set cameras inside of this cave and to see the otter activity during the day and night. So we set 3 cameras in each cave, and we left the cameras for 40 days. And we control the cameras in September and October. So all the cameras was removed in the first week of October. And so the station was reached by using the boat. This was a very useful tool for us to cover all this area in a short period. And related to the results, so as I mentioned before, we started to collect data. We did assessment of habitat caves in macro Prespa Lake. So this happened in July last year, and we visit this cave together with the administration of the national park. And based on the criteria that we used, so the vegetation present or absent in front of the caves, all the distance of the caves from the shore lake. Another criteria was the presence of the people that visited the cave. And so based on this criteria, we estimated that the habitat of each seems to be very good in the macro Prespa Lake. And of course, after that, we did training about the ecology and biology and monitoring methods. So the training was provided by PPNEA, increase the capacity of administration staff of National Park. So after that, we in the first week of September, we started to collect data, and so as you see in the map, the otter presence was confirmed in each monitoring station. So in the 25 locations that we did find and observed, we found it different types of signs led by otter, but mainly the Spraint was dominated, and also the track was founded, and so the otter presence was confirmed in each station of in Macro Prespa Lake. And so here is that can show the types of the signs that we found. As I mentioned, that was dominated by Spraint, and the Spraint was founded in this place that otter use to mark the territory, and they are quite visible on the top of the boat, on the top of the stones, rocks. So yeah, these signs were found in this habitat. And of course, we found the remaining food, as you see in the picture. So only the head of the fish was eaten by otter, and the body was left. And of course, the track and the resting place inside of the cave. So we found plenty of sprains led by otters. Even the sprains were very fresh. So there is another chart that shows there was a difference in the number of the sprain points in each station. And so the number of sprain points varies from 1 to 9, and the highest number of them was registered in station number 17, close to Golumboj village in Macro Prespa. And there is a difference in number of sprain points with fresh sprain found in each station and the period where the monitoring was conducted. So during the first week of September, there is also that we collected a high number of sprinting points in each station within the transect that we did in each station. So the data, we are not estimating the numbers of sprains in each station, but we are estimating the number of sprinting points within a transect, which is start from 200 meters to 1600 meters. Because when we started this study, it was very difficult for us to choose the right method, because it was a big debate based on what we have done in different countries. If you use the number of the sprains, you cannot come in the right conclusion about the density of otters, because the number of the sprain depends for different factors. And we combine the two methods that focus mostly in the sprinting points, number of sprinting points, than the number of sprains accumulation in one station, and the camera trapping method as well. And this is the result from Micro Prespa. So we had only 3 stations that we observed, and in each station we found signs that confirm the presence of otter in this lake. And no tracks and resting place or resting and remaining food left by the otter were found in Micro Prespa. This is, we think that is related to the type of habitat, so too much vegetation, the density and quite dense. So the movement of otter can be limited by this denser vegetation and can limit also to catch a prey. And also it was very difficult to find such of this place that you can find the signs left by otter. And this is the result that we obtained from a camera trap. So we set, as I mentioned, 3 cameras in 3 different caves, and we have a picture of otter. This is a picture of otter. So in the first picture, and also beside the otter picture, we collect other data. So from different species, wildcat was present and visited these caves too, and fox. But we are thinking now to identify more caves and to set more cameras just to collect more data and to see the otter activity or the structure of the family, so the number of individuals that use this, the caves and other data. And of course, the study, the survey is will repeat it again next year. So in the same study areas and in the same period. So we are collecting data, and after we'll compare the data that we collected in 2021 with the data that we'll are going to collect in 2023, and to see the difference in the number of the spreighting points, and to estimate an index and to see if there is a difference or about the trend of otter population. So to see, based on this data, we can understand how will be the trend of otter, but not to go and to estimate the density, because the method is quite limited and it doesn't give a full picture of the otter density. So yeah, the presence of the otter was confirmed in all monitoring stations in both Prespa lakes, and but we are next year we'll see how will be the conclusion about the trend, and the capacity of the staff of administration staff of National Park and interns, so was increased about the ecology, biology of and monitoring methods, and so it is very important that to continue or to collect data, not only in our site in Albania, but it's very important to have a full picture of otter trend population in all Prespa basin. It's need to do a transboundary survey between Albania, North Macedonia, and Greece. And we need also to work more with the protocol that we use, because all the data has been registered in the formal protocol that we used before to estimate the otter present, but not to estimate the trend of otter population. So we need to work and to update and to adjust this protocol. So thank you. I don't know if we have time. We have a video where a local from Prespa taken a video with 3 different individuals in macro Prespa. So if we have time, we can show this video to you.
Yeah, you can go ahead.
Okay.
if anyone has any questions in the meantime, if you put them in the chat, and I'll ask Bledi and Klaudja after he's put the video on.
I'm not sure if I share this video with you.
No, it's not on at the moment. You just need to, I think, share your screen because we're just seeing your video at the moment, like yourself.
Please do double sharing as in your screen. Ok. Está a nível funcional.
Posso pedir todo.
Ok, é legal. Deixa eu suggest. Nunca calma. Well, some Albanian in the background. We will probably put this on our social media page later on today because we got it yesterday night. So
oh, perfect.
We can just share it afterwards as well. So
yeah, that'd be great.
We won't take more time on this. Hello, Bledi Hoxha.
Okay.
Okay, that's perfect. There was a few questions. Really good presentation. Really informative about obviously your otters in Albania. One question I had personally was obviously PPNEA are doing research in Albania, but particularly the macro Prespa Lake covers North Macedonia and Greece. Are the organizations that you work with with Prespa Net, are they doing similar surveys in their parts?
I can answer maybe. At the moment, we are not doing the survey in all the 3 countries, but we have done it a few years back in 2017 jointly with our Macedonian partners. At the moment, though, we are working to establish an otter rescue team that is set in a transboundary scope, so that we can have a group of local reporters from all the 3 sides. But the survey is taking place only in our part of Prespa at the moment.
Perfect. There's another question from Kiwi, who's unfortunately had to leave. He asked, or she, sorry, they asked, is it possible to identify individuals from the camera traps, and from the 6 caves, 5 having otter Spraint in them? Are you able to know if they're different individuals or the same individuals within the caves?
Yeah, actually, if you collect data through camera traps, and yeah, of course, we can see the video with there are how many individuals there are for one cave. But if you try to compare this data with the data come from another caves, sometimes it's very difficult. And actually, it's the first time for us that we are using this method. But this is just to see how is the structure of the family of otter in this habitat. For example, how many individuals can find together, and yeah, to see there is a reproduction. We can, yeah, through camera traps we can collect data about to compare the presence of the species, reproduction, and so to see the structure of family. This works very good with the species or animals that have natural signs in the body, for example, for the Balkan lynx. So we can compare this picture and you can see how many individuals are identified in the area. But with otters, I think it doesn't work well to estimate the density.
Yeah, I would agree from our experience as well. There's certain species of otter, such as like the spotted necked otter in Africa and the giant otter in South America, that have neck markings, like quite unique neck markings, where the European or Eurasian otter doesn't really have that. So it's a lot more difficult to identify individuals in that respect, like kind of Bledi covered. If anyone has any questions for any of the presenters, I can direct them, and if all the presenters are happy to answer them. There is some varied questions on different aspects. For example, if Mohan is still available, a question just as you were leaving or just finished speaking was, is when you're doing Spraint surveys in relation to the 3 species, is the Spraint similar, or is it very different between the 3? Well, you've maybe not necessarily done Asian short-clawed otters, but between the smooth-coated otters and the Eurasian, is the Spraint quite similar?
So is this for, can I answer a little?
Yeah, that's for yourself, I think.
Yeah, actually, you know, like the Spraint that I use was more specific to the Eurasian otter, but I think it is a little bit different with the smooth-coated otter, because the shape, the texture, and some kinds of, you know, like a diet. Though I don't have a detailed study on diet, but the texture that is with the Eurasian otter and smooth-coated is a little bit different. And moreover, like Eurasian otter are found to leave the Spraint in some specific area, while the Eurasian otter, they go for different places. So I think there will be a little bit difference, but we need to validate with some specific study as well.
Yeah, I think kind of to follow on from that, from our last workshop in Malaysia, we kind of focused on the smooth-coated otter and the Asian small-clawed, short-clawed otter, and we found Spraint of both of these individuals. And again, they were quite different, mainly in relation to size. The size difference between the 2 otters is quite significant, and also diet, like Mohan suggested. The bigger smooth-coated otter, not exclusively but predominantly, eats fish, whereas the Asian short-clawed otter might eat sort of crustaceans, crabs and stuff. So you can see that in their Spraint as well. So yeah, there are differences.
And that's also my interest of the study as well.
Yeah.
A follow-up on that. Have you tried to distinguish them by smell? So I would be really interested in the other Asian otters. Do they also smell salty when you smell their Spraints?
From my semi-limited experience, I think without saying that it smells nice, the Eurasian has the most pleasant of the three. The smooth-coat, the short-clawed, and the Eurasian otter. And maybe Paul or I think Grace of IOSF might have a little bit more information in regards to scent that they might be able to give to you. And there's a little bit, I think they're coming on.
Hello. Some people describe Eurasian as being like jasmine tea, which I think is a bit excessive. I don't know what jasmine tea people drink, but I would say it smells sort of sweetish. It's not unpleasant at all. If you've smelt mink or something like that, it's disgusting, but Eurasian tends to be sweetish, fishy, very
salty.
distinct.
No, that's just in my experience. When you have something like a marten or so, where we have similar shape, a little bit smaller in the Spraint, but if you want to be sure and you don't have any unique fish bones or something, so that was just curious whether there's a difference in smooth coats or small claws.
Small claws smell horrible.
Mainly down to the diet. They obviously
eat
different things to the Eurasian and the smooth-coated.
Yeah, I think it's because the Eurasian eat more fish. I mean, they do vary, obviously, and the small clawed eat crustaceans and things like that. So that's probably got a lot to do with it. Yeah, but it's not unpleasant.
In my experience, the Eurasian in Portugal, they eat a lot of crayfish as well, especially inland, but they still smell sort of salty, fishy when you
Yeah.
Yeah, it's very distinct. Once you've learnt the smell of it, you know what it is.
Or could be like, can I add a few more? Like, could be like a Fred. Maybe the distribution, like the geography or some kinds, Eurasian otter in Nepal are more in the freshwater. And maybe like you have the Eurasian otter in some beach or the sea water. So it is itself said water in sea is rich with the salt, but in fresh water it is devoid of or some kind of salt, So maybe there could be some difference as well.
Yeah, absolutely. In regards to other questions, Ramesh has asked, firstly, thanked everyone for their presentations, and they asked this is kind of directed at Paul, but I'm sure Eleanor or Fred could also answer, or anyone could actually answer, sorry. What is the best method for estimating populations? As you said, Spraint cannot be correlated with population.
I suppose it's a DNA work, but if you're looking at coastal otters, like I did my research on, there's a correlation between active otter holts and resident females. So if you count the active otter holts, you can work out a relationship and work out a population. But it would be very difficult to do in a freshwater river system.
Yeah. And Teresa Mason, thanks, Paul. Teresa Mason asked to all presenters, there's a couple of layers to this question, but does anyone use environmental DNA testing of rivers? And then, is this the way for the future, or is there no real replacement for surveying? And finally, do you think climate change is affecting otter populations yet? The latter of the questions, Teresa, is an interesting one, and I'll let our speakers emphasize this. But if you're attending the later presentation, it's obviously based on a different species to the ones we're kind of focusing on here, but there is a little bit more information on one of the presentations in relation to climate change, just as a heads up for anyone that might be joining later and might be thinking about it. But I'll open those 3 questions to the presenters as well.
eDNA has been used. There was a guy did it in England. I think it's still quite early days, but it is going to be more important, I think, as time goes on.
Yeah, there's a PhD student at, I think she's Salford, she's called Holly Broadhurst, who's doing some work on eDNA as well, estimating from water samples. I think my understanding is that at its current sort of development in the technology, it's still going to be allow you to have an estimate of distribution rather than any more of those detailed questions that people desperately want to know, is how many otters are there and that sort of thing. But methods, particularly genetic analysis, are developing all the time, so who knows what will be possible in the future. And one of the problems with DNA analysis from otter spraint in the past has been low success rates. Well, sort of still a problem, but again, there's people developing new techniques and improving that all the time. So I think there's possibilities. I think the main limiting factor again is funding, though, for any of this work. It's possible to develop methods, but if the analysis of individual samples is still really expensive, there just isn't the money around to do that work, however much we'd like to know how many otters there are. And just briefly on the other part of that question about climate change impacting otters, in the UK, I would say probably the main impacts are the impacts on their prey species. So we know, for example, there's climate change impacts on some fish species if the river is too warm over winter, and that's impacting fish populations, and of course that will impact otters because they need to eat.
Perfect. And then kind of to follow on, Anne, who is one of our attendees today, said the Mammal Society conference had speakers on eDNA, and they implied that otters are difficult to detect via eDNA, which is an interesting addition to anyone who has experience or the question in regards to eDNA.
Yeah, and similar to in their Spraint, you know, you can take the droppings from another species and have really high genotyping success rate, and then for otters it's really low. So they're just tricky creatures.
They are tricky. They're elusive in every way possible.
There was an interesting poster at the IUCN Eurasian Otter workshop where someone looked into audio data. So they set up audio mod and looked into bioacoustics. And in this poster, it was also, I think, it was possible to identify individuals. So the way I see it is in the future, a combination of tracking, but also machine learning of various sensors could definitely be something of interest. But as Eleanor said, it really depends on the availability of funding. So goal when developing methods should, in my opinion, be something that can be cost effectively be widespread, and then really depending on the region what's available and most applicable, and then just see a mixture of sensor data and ideally artificial intelligence assisted analytics, because also you need to keep in mind that analyzing can take a lot of time if it's done by an expert, and if that time can be re-navigated to other areas, then more data could be collected, which is necessary in my opinion.
Yeah, I'd agree. Is there any questions that people might have for any of the speakers, or even for myself if you want to ask a question? I will give them a few minutes. Yeah, like you say, Frederick, I think if you can get AI, then it also, not that we are necessarily going to make errors, but humans make errors, and if you can get the AI with maximum data, you reduce the error rates.
Yeah, but also on camera data, for instance, like I know it's still other groups are developing on that, but there's groups looking into detection of like classification of camera data, for instance, because it takes also time to run through hours of camera material. And I read of a project looking at grizzly bears. They tried to do like a facial recognition of grizzly bears in order to individually identify whether this is going to work for otters. If a human can't do it, then a computer most likely won't be able to do it either. But this is, in general, it in my opinion really depends of where you are and in what works good in your region, and then just take an appropriate tool out of your toolbox. But it's important to cross-validate these tools among others so that you have comparable data over regions.
Yeah, definitely. Well, I think in regards to the questions, everyone's very happy, and all the questions have been answered. So I will take this opportunity to thank Dr. Eleanor, Dr. Paul, Frederick, Mohan, Klaudja, and Bledi for their time, their expertise, and their enthusiasm. Thank all of the attendees who have taken the time out of their morning to join us and listen to all the amazing talks we've had today. And finally, for anyone that's joining this afternoon, we'll see you in a few hours. For any of you that aren't, enjoy the rest of your World Otter Day and have a fantastic day moving forward. Thank you, everybody.
Bye.
Thanks very much.
Bye.
Bye-bye.
Very
Thank
much.
you.
Thank you.
Thank you.
Bye.